My name is Molly Carrig, and believe it or not, my job in lab research is similar to that of a marriage counselor. A marriage counselor seeks to answer questions about how two partners get along. What made their connection good at the beginning? Why did the two people get married? What constitutes a good connection? How does a good connection form, and what steps are involved in that process? And what are the consequences of good and bad interactions? The process of marriage counseling really is a drama, filled with victories and setbacks, problem solving, and celebrations. Both marriage counseling and my research represent challenges of perseverance. It's working to answer that key question and find the solution. But as it turns out, there are thousands of tiny things going on behind the scenes, all of which are required to answer that one fundamental question. A marriage counselor wants to figure out how to foster a connection between two people. In my research, I want to figure out how to foster a connection between two proteins. It might seem like the interactions between microscopic proteins would have microscopic effects, and of course, in some senses, they do. But these microscopic effects play out on a very large role in the body. Just as what happens in a couple's private life often affects many, many others in a giant ripple effect. Think about the disease that 1 in 3 people in this room are likely to contract during their lifetime: cancer. Statistics certainly aren't necessary to convince anyone here of cancer's widespread and devastating effects. I would guess that everyone here has already been affected by cancer in some way. But how does something in our so well-tuned bodies go so wrong that our body ends up attacking itself, with healthy body cells being destroyed by cancer cells, which are in fact your body's own cells? It turns out that some cancers can be caused by faulty connections between two proteins, a couple that simply will not get along. As I mentioned, cancer cells are from your own body. They do not enter your body from an exotic plant, a diseased insect, or even another human being. So the problem lies all the way back in your DNA, which codes for those very cells. DNA is the genetic code of the body, and it gets a lot of hype as that double helixed holder of all information. And that's true, the DNA does hold a lot of information. And sometimes DNA damage by elements such as UV exposure can lead to cancer. However, the DNA can code for everything perfectly, and information can still be severely corrupted. The DNA code can be read incorrectly by the mechanisms that come along to read it. This is where my two microscopic but very important proteins come into play. During the process of DNA replication, proliferating cell nuclear antigen, pCNA, acts as a sliding clamp, moving along the string of DNA. In some cases, it will bind to another protein, chromatin assembly factor 1, or CAF1, that blue protein there. When these two proteins bind, CAF1 sends out a call for something called a histone. Once the histone arrives, sections of the DNA wrap around that histone. They cling together so tightly that nothing can get in between them to read the DNA. Just like the two proteins that bound so nicely, now the DNA and the histone are inseparable, the very best of friends. So when the machinery that's in charge of reading the DNA comes along, it simply cannot get between the sections of DNA wrapped around the histone. That whole unit is too bound together. So when my two proteins bind, that area of DNA is not expressed. Imagine if, say, two friends were so close that when a newspaper interviewer came to talk to one of the people, the friends were so constantly engaged in conversation, and even standing so close together, that the interviewer had no chance to learn about that one person. And although this may sound like a bad thing, it's actually a much needed tool in the body. This exclusive friendship makes sure that not every single bit of our DNA is expressed every time. Think of DNA like one of those instruction manuals that come with computers. The ones that are about 20 pounds and end up on a shelf for use as a doorstop. These manuals contain thousands of pages of information. Information that should really only be used by certain people and at certain times. Some sections involve cleaning your hard drive. Others talk about screensavers, internet connections, syncing calendars, and changing your music settings. Some sections should really only be read by the Apple staff, who unscrew the bottom of your computer and deal with that delicate, maybe radioactive part. Imagine if every time you opened up your computer, you did every single action in that manual, front to back. Did every single thing it told you to. Your computer would, at the very least, shut down and overload. Maybe even implode. I've never really tried it myself. This is basically what would happen if every part of your DNA were read every time it was expressed. Our DNA contains a lot of information that we don't want to use every single time. The choosing of which DNA to use is what makes our heart cells, and our brain cells, and our skin cells. However, if my two proteins, PCNA and CAF1, don't bind properly, this can be just as explosive as a nasty divorce. If they don't bind, that histone that we talked about before that the DNA wraps around, it never shows up. And then that area of the DNA is packaged very, very wrong. This wrong expression can be a cause of cancer, because on its most fundamental level, cancer occurs when cells replicate abnormally and eventually uncontrollably. All of this incredible information about cellular level bonding has been discovered by scientists over the past decades. However, there's an important, critical, crucial missing link. We don't know any more about how these two proteins bind than we do about how a random couple off the street fell in love, what mutual experiences and characteristics made them perfect for each other. So that's what my lab is trying to figure out right now. How do these two proteins bind? Because while it happens on a microscopic level and in a matter of an instant, when it goes wrong, there are absolutely disastrous consequences. The process of research itself is far more dynamic, exciting, and challenging than I'd envisioned before beginning my undergraduate work. When I was in high school, I thought research involved putting on a white lab coat, looking under a microscope, maybe mixing some chemicals, definitely a lot of waiting, having a eureka moment sent down by God with a great discovery, and then writing a really long, really boring paper about it. However, it turns out that I don't get to wear a nifty white lab coat on a daily basis. I've never once used a microscope in my lab. And while I do mix some chemicals, they have never yet caused any explosions. Lab work is like the investigation phase of a marriage counselor, and it definitely requires a lot of perseverance. A marriage counselor conducts surveys and diagnostic tests to get to know the personalities of each partner. In the same way, we try to get to know the personality of each protein. To get to know our proteins, we first have to isolate them. One of the steps in this process involves using a purification column. We put a solution that has the protein we want, along with a lot of other cellular junk, into this column. We pour it right on in, and through the process, we filter out only the protein that we want. Sometimes if the goo is super thick at the beginning, I've spent whole afternoons sitting in front of the column, watching it drip. Slowly. Drip. 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 Because if the column runs dry for even a single second, our proteins get very, very upset. I think back to one afternoon in particular, where the proteins were becoming solid in the solution, something we definitely don't want to happen. To try to fix this problem, I set up the apparatus on the floor of the lab fridge, because proteins like the cold. And for the rest of that afternoon, I was sitting on the floor next to the fridge, cross-legged, slowly dropping in solution, and making sure that my very hastily built apparatus in the fridge didn't collapse. My colleagues laughed along with me, partially at me, and tripped over me quite frequently, as I babysat those proteins on the floor for an entire afternoon. So after we've isolated these proteins, after a marriage counselor has assessed the personalities of each partner, she now needs to figure out how those proteins bind each other, how the personalities complement each other. As with marriage counselors, after isolating our protein, we need to figure out how they fit together in binding. One of our approaches to protein marriage counseling is testing different topics to see how the couple reacts. You might venture to discussing mutual friends and see whether that makes the couple get along better or worse. Maybe even bring a strong friend of one partner to the session and see if that helps interactions go more smoothly. If that went downhill, maybe a marriage counselor would bring up a trip to Paris and see if that makes the conversation better or worse. Maybe a discussion about compassion or honesty will remind a couple about shared values that brought them together in the first place. With our proteins, we add protein fragments, mutate segments, and even delete sections, seeing if that makes the binding better or worse. One of the ways we've messed with pCNA, one of the proteins, is to bring in a friend, another protein called Sumo. So this ring here is pCNA, and these little sections that we add on are called Sumo, a small ubiquitin-like modifier. We know that this Sumo exists bound to pCNA in the body, and we think that there might be an area on CAF1 that binds Sumo really, really well. We call it a Sumo interacting motif, or a SIM. To see whether or not this region is a SIM, we add the Sumo onto the pCNA. If the region is a SIM, then the Sumo in pCNA should bind really, really well to that CAF1. And if it's not, there should be no more binding than usual. So to run this test, we use these pretty plates right here. And how this works is we put one of the proteins, say pCNA, into the well. Then we add CAF1 and rinse it out. If the two proteins bind, the CAF1 sticks in there very nicely, and you have your two proteins in the well. If not, the CAF1 gets rinsed out. Then we add a molecule that will cause CAF1 to turn orange. So once we add that to all the wells, if it turns orange, that means there's CAF1. It wasn't rinsed out, and there was binding. No orange, no binding. So I wanted to show you what the results of these tests actually look like, instead of just throwing a graph at you. Also, I think they're very pretty. I quite like the bright orange and the gradients. But when we saw these tests, we get very excited. When it turns orange, we say it lit up like a Christmas tree. And when I got the results of this test, I personally jumped up and down at the lab bench. And then I had to take a moment and laugh at myself for the absolute nerdiness of that moment. But it's true, we get really excited when we finally get data, especially since it takes a long time and doesn't drop down from God in a eureka moment. So let me explain this test a little bit. The top row is our positive control, and that's just CAF1. It lit up, which is expected because the orange means that there's CAF1 there. Great. The bottom two rows are the negative controls. Those are just pCNA, and they make sure that we don't have any false positives in our data. They did not turn orange at all, which is great. It means that what we're seeing for binding is actually binding. So I'll show you the graph version of this. So it turns out that when we add the Sumo to pCNA, this top blue curve right here, it binds CAF1 really, really, really well. For comparison, regular pCNA and CAF1 is this middle curve here, and those negative controls are at the bottom. This makes us think that the CAF1 piece does in fact contain a Sumo interacting motif, a SIM. So from this, we compose that the region is a SIM, and that its target in the body is simulated pCNA. Next, we're looking at using X-ray crystallography to shoot a beam of X-rays at our proteins and figure out if we can get an actual picture of what's going on between them. This would be like if a marriage counselor walked up to the couple's window of their house and just filmed them for hours on end. This would be the holy grail. This would be the actual picture of what's going on between our proteins. In the meantime, while we're working on that, I'm also running other tests to see if we can better quantify the interactions, put numbers on them. Like right now I'm trying to get thermodynamic information about the amount of heat released when these two proteins bind. Here's the thing though. Right now we have a supported hypothesis. And that's great, but it's just a half step towards one piece of a 1,000-piece puzzle. We still have a lot more work ahead of us, and each discovery leads to 1,000 new questions. Just like partners encountered in marriage counseling, proteins are incredibly complex, and their personalities are difficult to unlock. We're hoping that if we ask enough questions about the proteins and keep testing to see what makes a good connection, we can figure out exactly how they bind in the body. This is an exciting challenge ahead of us, and perseverance will be a little easier to muster when facing these 1,000 questions, given our exciting lead. But remember, it's not always that exciting. Sometimes perseverance looks like sitting on the floor of a lab for an entire day. Just the other day, I was running an experiment to try to find those heats of binding that I mentioned earlier. And my partner and I, after triple checking our calculations, spending about 12 hours in lab, picnicking for lunch, dinner, and midnight snacks in the hallway of the science building, we got inconclusive results. Research isn't always explosions and bubbling beakers, but I think I'm still a happier and more fulfilled person because of my time in lab. And it's not that knowing about these two proteins in particular makes my life better on a daily basis, though I do think they're pretty cool personally. I'm better because of the critical thinking and the problem-solving skills, those tools of perseverance that research has allowed me to develop. I'm able to see long-term, to pick out the hope that drives us. And I'm supported by a remarkable team of my peers and my research professor, people who are equally as passionate and optimistic, goal-driven, and determined as I am. Our team in lab gets to face these challenges together, the exciting and the frustrating, the novel discoveries, and the absolutely baffling roadblocks. The lead we have gives us hope, and the hope inspires continued perseverance. Hope gives us the strength to ask question after question, to be certain that with a determined team and continued perseverance, we will know how these two proteins bind. And then, not only can we work on making sure they bind more strongly, but we ourselves will come out on the other side as stronger individuals. Thank you.